We're going to start out by finishing up with our endocrine system, reviewing what's going on in the absorptive and the post-absorptive state. So if you want to grab your notes on that and look over it real quick, about to bring up a series of questions on it. Okay, we're going to do this in short quick form. Okay, well, everybody's with us who's joining. If not, follow along as we go through these questions. I'll see some more people joining, so I'll give you another second. Take a look. Okay, so we're going to start. And you're telling me what hormone is responsible for regulating blood glucose in the absorptive state. Okay, great. Yeah. Our two hormones we're interested in are insulin and glucagon. Insulin is corresponding to our absorptive state. Okay, so where's that insulin coming from? Okay. Insulin is coming from our beta cells. Glucagon is coming from the alpha cells. The exocrine cells are for enzymes. They're surrounding the islets of Langerhans. They're not involved in this endocrine hormonal function. What does insulin do and not do? All right, this time we're just going to run a bit fast for this. So last time, when we were talking about absorption and absorptive, we were categorizing our two states as either building stuff up or breaking stuff down for the most part. So insulin during our absorptive state is the building stuff up hormone. So in order to do that, we're going to pull glucose into cells. Right? We got to do that because we just ate stuff. We're going to make fats. We're going to make protein. And we're going to stop breaking anything down. So we're not going to be breaking down glycogen at this point in time, because this is all about building stuff up. So glycogenolysis was breaking stuff down. Finally, when are we going to be releasing this insulin? So we got almost an even split. So when we're thinking about what's triggering our absorptive and our post-absorptive state, I'm going to do a little drawing, right? Okay, so this is going to be my intestine here. We're going to have our bloodstream above it, okay? And then I'm going to put some cells above that. So this is our tissue. Okay. So what's happening when you eat food is you are absorbing the contents of that food. So one of those things that you might be absorbing is glucose, right? So if you eat something sugary, it's got some glucose in it. Glucose starts out in your intestine, right? So that's step one. And then we're going to go through in A&P2 all the processes that control this, but glucose first gets absorbed into your blood, right? So that first step is just happening through digestive processes. So the way that your cells know, right, that you've just eaten is once you have that glucose in the blood, right? So once you have the glucose in the blood, now we've got the circulating products of digestion in our bloodstream, and we have to do stuff with that. Right? So when we want to produce insulin, right, that's going to be our initial trigger, right? Insulin is part of the absorptive state, okay? All right, so we're building stuff up that we've just absorbed. But first it had to get into the bloodstream. So if we were thinking about glucose levels, right, what would happen with our glucose levels? We're going to track before and after you eat, right? So here's where you eat. And this is going to be over time. You're going to have lower levels of glucose before you eat. You're going to eat. Your body's going to absorb that glucose, right? Insulin is then going to get released, right? Because we want to bring that glucose back down, right? So here's our insulin release. So that'll pull our blood glucose back down to normal, okay? So here we're tracking an imaginary glucose level. So the way it's doing that, in part, is by controlling these tissue cells, right? So in the presence of insulin, right? So when there's insulin, what we're going to do in part, right, on the previous slide, there was that mention of increasing glucose uptake. So what we can do in the presence of insulin is add some transporters, some proteins in the walls of our tissue cell, or the membranes of our tissue, right? Allowing more glucose uptake, right? So glucose goes into the cell. If we had glucose in the blood, release insulin, but now we're pulling glucose into those cells. That's what's pulling us back down, right? So the glucose isn't disappearing from your body. It's going into the tissue cells for use to produce ATP through our cellular respiration, or for like the liver or muscle, maybe we're building up some glycogen molecules, storing that for later. But that's part of the purpose of insulin. This is part of the problem. You have diabetes and have to give yourself insulin injections, right? You can imagine if you don't have insulin, right, your sugar is going to go up when you eat. And maybe it'll drop a little, who knows if you use some, but right, like it might just keep going up depending on what you do. It doesn't come back down, right, without that insulin telling your cells to respond here. So one of our triggers for that release of insulin is the rise in blood glucose. A result of insulin release would be decreased blood glucose. Congratulations to several of you. All right. So getting back to that glucose. Right. So that was actually a great segue because we ended out on the teaser about blood glucose levels. Right. So glucose is something that we're trying to regulate through homeostasis. It's a regulated variable. We're trying to keep it in this normal range, right? So we are trying to keep it in that 70 to 110 milligrams per deciliter range, right? And deviations outside of that range are going to be dangerous, though in different ways. And so in hyperglycemia, we categorize that usually as substantially above our normal range. So we're going to start it at like 140 milligrams per deciliter. This is too much glucose. So this is over time going to kind of wear on your organs. Right? Hypoglycemia is also a problem, in part because your nervous system relies mostly on glucose to fuel all its processes. So like your brain needs glucose in addition to your nerves out in your body, right? So this is going to be kind of an immediate danger if we're affecting your brain. You have this hypoglycemia, this drop, this precipitous drop in glucose. Out, right? Your body functions might stop working as your nervous system stops working. So we define hypoglycemia as below about 60 milligrams per deciliter. We want to maintain our blood glucose levels in this normal ideal range. Usually you'll see this actually ballparks, I think like around 100 when you see it in the textbook as a glucose level, but we got a normal acceptable range for blood glucose. So it would depend a little bit depending on how long ago you ate, how much insulin, how much glucagon are operating in your body at that time, right? So when it goes up, your glucose levels go up. You want insulin to tell the cells in your body, pull that glucose into the cells and do stuff with it, store stuff, use it for energy, right? If your glucose levels are dropping too low, you want glucagon to be released because you want the cells to stop pulling in glucose. You want places like your liver to start making new glucose out of other stuff. Anywhere you have glycogen, you want to start breaking up that glycogen to release the individual glucose subunits for energy. Put that into the blood, try and raise that glucose a little. You want the cells in your body in general, if you're not in the nervous system, to try to use proteins, try to use lipids to get energy. We know lipids, triglycerides are really good store of energy because they can really densely pack ATP into those bonds in those hydrocarbons, those carbon-hydrogen bonds. We briefly mentioned beta oxidation because of how many of those are packed into a fatty acid. We get a lot of energy out of that if we're using fat for energy. So here's what these pathways would look like in the example figures from your book. Right? So what we're looking at here is regulation of these glucose levels. So say you have glucose go up, right? So that's our stimulus, our glucose going up. This is how your pancreas knows that it's time to secrete insulin. Right? So the beta cells in your pancreas, ones that are responsible for insulin production. So they secrete insulin. Secreting insulin, meaning they're making it and they're putting it into your bloodstream. Now that insulin is circulating around all throughout your body. So it's going to tell most of your tissues to pull glucose into the cell. So we can immediately see that that's going to drop down glucose. If we took it into a cell, it's not in your blood anymore. We also have effects in the liver and the muscle. Right? So one thing, when we pull in glucose, we can link together glucose into glycogen. Right? So we're going to see that happen in the liver and muscle. We're going to stop breaking down glycogen, that glycogenolysis. Right? We don't want to break it down because that would release more glucose into the blood. That wouldn't be helpful. And your liver is able to make new glucose. We don't want it to do that. If our problem or our thing that we're noticing is that we have raised glucose levels, we want to bring the glucose levels down. So we're going to stop making new glucose. That gluconeogenesis, creation of new glucose, should stop in the observative state under these conditions with the control of insulin. Questions about this process? So let's do it for glucagon then. All right. So glucagon is just the opposite. So glucagon is for post-absorptive state. So we are going to be trying to keep our glucose levels up. Right. So our initial stimulus for releasing glucagon from our alpha cells in the pancreas is going to be when we notice a drop in glucose. Okay. You ate a while ago, 4 or 5 hours ago, gone on a run, you've got to work, you've got to class, you've been using up the glucose that was in your bloodstream, you're making stuff out of it, you're burning it for energy. Now there's not much left in your bloodstream. So now it's going to be time to release your glucagon. So those alpha cells increase our secretion of glucagon. So making the glucagon, releasing it into your bloodstream, circulates throughout your body. Okay. So when it gets to the liver, tells you, okay, we don't have enough glucose in the blood, make us some new glucose. Right. So that's that gluconeogenesis, right? So making new glucose. Right? And another way we can get glucose into the bloodstream is we can break down the glycogen. So the glycogen is a bunch of glucose glued together. So when we break down glycogen, hopefully that'll raise our glucose a little bit, right? Not going to have as much of an effect as actually eating, right? That would be the way to really get your sugar levels back up. But we're going to try and make a little glucose. Okay? And on the other side, we are going to be trying to use other sources of energy throughout the body so that we can save our glucose for the nervous system. So the glucose sparing is because our brain needs glucose, right? So when we say it's spared, that means brain is using it, right? Okay, so we're saving it for our nervous system. Okay. So everywhere else, we want to use other stuff. One of our best sources of energy are our lipids, right? So we're going to increase how much we're breaking down lipids. That lipolysis is the breakdown of lipids, in particular our fatty acids, right? So our fatty acids are going to get into the plasma. We can then take those fatty acids into other cells, break them down, use them to make ATP in other ways, which means we're saving any glucose that we do have left in the bloodstream, specifically for the nervous system. And that's our big goal here. Keep your brain going, keep your body going, even in these post-absorptive states. The post-absorptive state could be really long, right? You don't always eat every 2 hours, right? So hopefully when you're studying, what you want to focus on, right? You want to be able to associate insulin and glucagon with their appropriate states, right? So we just talked about insulin with the absorptive state, glucagon with the post-absorptive state. We happen to know that some of our nervous system effects go along with absorptive and post-absorptive pretty well. So we have our sympathetic nervous system, epinephrine, kind of that fight or flight part of the nervous system. So that's going to be more like our stress response, our post-absorptive kind of state versus our rest and digest is the parasympathetic. So that's more likely to be happening during that absorptive state. And we want to make sure that we can tell apart all these many G words in this unit and appropriately associate them with either insulin and the absorptive state or the post-absorptive state and glucagon. Cool. So in summary, that's pretty much finishing up, actually that is finishing up our endocrine section of this unit. So what we've done is we've gone through our different primary endocrine organs. So the ones that are their main job is to release hormones, so signals into the blood that do stuff elsewhere. Right? So we spend a lot of time talking about the hypothalamus and the pituitary. That's one of our really important topics for this block. right? We want to understand tropic hormones and that interaction between especially the hypothalamus and the anterior pituitary. We have lots of anterior pituitary hormones. We have that kind of tropic flow because we have hormones from the hypothalamus affecting the release of hormones from the anterior pituitary, and then potentially affecting other endocrine organs past that as well. In particular, we focused in on the thyroid gland and the thyroid hormones, as well as the adrenal gland and that adrenal cortex, talking about the release of cortisol. So we do want to know the existence of these other primary endocrine organs and the other anterior and posterior pituitary hormones. Those are the ones we focused in on and spent more time with. Okay. Secondary endocrine organs exist. They're organs that have other jobs that are potentially more important than their hormonal signaling. So the heart, the kidneys, the GI tract, the liver, in all sorts of places, as well as the gonads. And here's a chart also from your book, charting a bunch of the hormones that we discussed. So this is in chapter 21. So you can see that we talked especially insulin, glucagon. We talked some about epinephrine, talked about thyroid and cortisol. We didn't focus so much on that growth hormone. Okay. So what you want to be able to tell us is where that's coming from, right? So where does the hormone come from? You want to be able to tell us, classmates, whoever, why you'd be releasing that hormone. These ones that we've listed here are ones that have some effect on metabolism. So you might relate that here. Really, when we think about glucose, I want you to be thinking about the insulin and glucagon, but we have other effects as well. So questions about the endocrine stuff before we move on to our next packet, which is the somatic stuff. Okay, here we go. All right. Yeah, okay. Let's remember, so jot down for yourself some of these terms. So we want to remember what it means. Something is afferent versus efferent. We want to remember what EPSPs are and what depolarization is, because these are going to come back as we think about this somatic nervous system. I'll give you a minute or two to jog your memory, and then we'll get rolling. To use these words. We're going to start with these first two. So the slide's going to come up in the picture on the next slide, but I'm going to draw this for you so we're talking together, following along first. So remember, when we're talking about, not a good C. When we're talking about the nervous system, we can divide between the central nervous system and the peripheral nervous system. And we're talking about the peripheral nervous system throughout this unit, really, right? So when we were talking about central nervous system, brain, that was really in our last unit. Okay? So we know that we have afferent nerves, the ones that come into the central nervous system. So part of the peripheral nervous system coming in. So those are our sensory nerves. So that's afferent. All right. which means that efferent is when we're coming out of the central nervous system. As we come out of the central nervous system, that's our efferent or motor sort of branch, right? So the part that's doing stuff, having an effect. Efferent, coming out of the central nervous system, right? Then we have another branching point. So we've been talking to this point about the autonomic nervous system. We talked about the autonomic nervous system, which had the sympathetic and the parasympathetic branches. We'll label those, but together, those two branches, we're going to our glands, our heart, and smooth muscle. So cardiac muscle, smooth muscle. And we are now locating ourselves on the other side of this efferent branch of our nervous system. So now we're going to be thinking about the somatic nervous system. So the somatic nervous system is the opposite of the autonomic nervous system. And we're going to be able to tell the difference straight away by thinking about where we're going, right? So our effector organs, we're going to be thinking about our muscles, specifically our skeletal muscle. So skeletal muscle, the muscle that attaches to your skeleton. So all the named ones that you've been learning in lab would be somatically innervated skeletal muscle. Okay. Which means that this is going to be really the first part of the nervous system that we see that has to do with voluntary control, things that you can actually control on purpose, versus everything with our autonomic nervous system really had to do with homeostasis, maintaining regulated variables, not stuff you're going to be doing on purpose. That autonomic nervous system stuff was all really kind of automatic, right? Involuntary. So you should be practicing in this unit, like checking this chart out, make sure that you can understand the branches of the nervous system and tell them apart. I'm not going to ask you to tell the differences between all the different types of the receptors for now, but you do want to know that we have sensory information coming in through that afferent division, our motor commands, all our commands really coming out through the efferent division, breaking that up based on where our nerves are going, skeletal muscle for somatic, smooth muscle, cardiac muscle, and glands for autonomic. And we spend a long time telling apart our parasympathetic and sympathetic divisions of the autonomic nervous system, all their different neurotransmitters, all their different pathways, all their different effects. So you can see that the somatic nervous system is a little simpler, doesn't have divisions within it. This packet is shorter. That's what we're doing right now. And it's going to be leading us into our study of muscles themselves. We're going to start out by understanding how somatic nerves work. So when we talk about somatic nerves, we're really going to be talking about motor neurons. We're often going to be referring to them as motor neurons because they're going to skeletal muscle, right? They're going to be giving motor commands, telling you to move. So motor neuron, you may have seen this described already in one of your labs, but in case you haven't, in case they changed those, I'm going over it again. All right, so our motor neurons are coming from the ventral horn here in your spinal cord. Okay, they're going out through the spinal nerve, and they're going to come down to your skeletal muscle. Okay, we're going to see that there are many motor neurons that go to the same big named skeletal muscle. So we're going to notice right now just that skeletal muscle is made up of multiple muscle fibers. We'll get into that later. So our neurotransmitter for all of the somatic nervous system is going to be acetylcholine. Right? So we've seen acetylcholine before. We know that acetylcholine was responsible for all that communication between preganglionic and postganglionic neurons throughout our autonomic nervous system, right? That was one of the places where it didn't matter if you're a sympathetic nerve or parasympathetic nerve. You're always using acetylcholine if you're going from the first neuron in the chain to the second neuron in the chain. So acetylcholine coming back here in the somatic nervous system. And specifically, we're going to see that acetylcholine in skeletal muscle is going to have a specific receptor type associated with it. And it's going to be that same receptor type too that we saw in the connection between our preganglionic and postganglionic neuron. We're going to see a nicotinic cholinergic receptor. These ones work fast. We'll often refer to them, kind of associate them with those ionotropic receptors that we talked about in our last unit, right? So they're really just going to quickly kind of open ion channels, work fast, versus our other type of cholinergic receptor was the muscarinic cholinergic receptor, linked up to a G protein. That's what you would see like on the smooth muscle, on the cardiac muscle, on a gland, coupled to that G protein, which means it can have more very different effects. When we see these nicotinic cholinergic receptors, what it means is that we're trying to set up an action potential, right? That's why I asked you what an EPSP or depolarization is, right? So when we are sending a somatic nerve to a muscle, we're trying to contract that muscle. And the way we're going to do that is really through an action potential. So we're again going to see ion movement, depolarizing, helping us depolarize cells, right? So depolarization is when we see kind of spikes in that resting membrane potential. We go up towards threshold when we're talking about our neurons in the brain. And so we're going up. And we also think of that as excitatory, right? So we had our EPSPs, our excitatory postsynaptic potentials. And that's going to be analogous to what we're going to see here in the communication between a somatic nerve and a muscle cell. So here we're looking at a variety of motor units. So we can see 3 motor neurons, 1, 2, and 3. So we can see that they are each going to muscle fibers within a named muscle. So here you've seen in lab how muscles look kind of thready, right? So it shouldn't be a surprise that they have fibers within them. Right, so any given motor neuron is going to start out as one neuron with one body here in the spinal cord. As it comes out, it may have branches, right? In fact, it will have branches. So here, as I'm tracing motor neuron 1, you can see it branches into 3 here. And you can see that each of those 3 branches goes to a muscle fiber, right? So we've gone to this muscle fiber, we've gone to this one, and we've gone to this one. Okay. So we can see this association between motor neurons and muscle fibers. We can see that one motor neuron starts as one motor neuron, but can go to several muscle fibers. If we look down the length of any one of those muscle fibers, right, you're going to see that we only have connections going back to one place, right? So we don't have any muscle fibers where you have joint innervation from motor neurons 1 and 2, right? They're always just linked up to one of those motor neurons, right? So every muscle fiber goes back to just one place in the spinal cord, one motor neuron, but any given motor neuron is going to multiple muscle fibers. That's what we have for our structure of these neurons. All right. So when I get to the muscle fiber, we're going to have what we call a neuromuscular junction, right? So neuro for the neuron, right? Muscular for the muscle, junction because they're coming together, right? So neuromuscular junction is like our modified version of a synapse, but now we're synapsing on the skeletal muscle. Okay. So we can look at both the neuron side, what we're starting with, and then the muscle side. Okay. So what we're going to be doing we're going to be activating that motor neuron first. And so in order to send a signal out to your muscle, you're first going to have to trigger that motor neuron. So the motor neuron is going to fire when it sums up all the little potentials, all the little signals coming into the motor neuron and reaches threshold for the action potential. Okay? So we have some signals coming in that are excitatory to that motor neuron. We might have some inhibitory signals coming into the motor neuron. We're trying to figure out if we get to threshold. Right? If we get to the threshold, we send off an action potential. All right, so that's what we care about. We send off an action potential. So we triggered our motor neuron to fire. Motor neuron fires. That action potential heads from the axon hillock down the axon towards the end of the motor neuron. Here's where we have our first kind of specialized terminology for our neuromuscular junction. We are going to go down the muscular junction, down this neuron, and get to a motor end plate, which is going to have an end plate potential. This is a little bit of different terminology. Okay? We'll take that when we come back and look at the thing as a whole. But basically, that's what's happened for our axon terminal linking up to, say, a dendrite of the next neuron in a chain. Now we're going to have a motor end plate and an end plate potential. We'll take a look at it. So just like in our other nerves, we're going to see that when we release a neurotransmitter, okay, just like throwing a ball, you're going to need something to catch it. So on the muscle side, we're going to have a receptor. So on the muscle fiber, on that neuromuscular junction, you've got a receptor for acetylcholine, because we know that acetylcholine is the neurotransmitter involved in the somatic nervous system. So we're going to have some more specific terminology here as well. So we're going to see a terminal bouton related to this on our full image of the neuromuscular junction. We'll see that in a second. And that motor end plate coming together. Basically, that's our combination that forms our new version of a synapse specialized in this neuromuscular junction form. So we're always releasing acetylcholine here. We're therefore always going to have a cholinergic receptor. Specifically, it's a nicotinic cholinergic receptor. Going to let ions flow through it when we have that acetylcholine bound in. Okay. And these synapses, these neuromuscular junctions, these are basically always going to be excitatory. There's really only commands from those motor neurons telling you to contract, right? It's just when the signal fades away that you relax. Right? So whenever you send an action potential down that motor neuron, you're telling the muscle to contract, to move something. You're never sending a signal that's inhibitory telling us to stop. That's just when we have our signal fade away. Okay, so we're always going to have these be excitatory. So we're always going to have these excitatory end plate potentials. And we're going to use an enzyme to break up our acetylcholine at the end, our acetylcholinesterase. So I think next we should have our full picture. Okay, so that's how we're kind of comparing. So let's take a look. So we have our motor neuron up top here. Right, so our motor neuron coming up, down, we've triggered an action potential somewhere up in the central nervous system. We don't know where. We have our signal that we want to contract the muscle. So our action potential is coming down. That's a wave of depolarization. So now we are getting to this terminal bouton. Our terminal bouton, so like this button at the end. So terminal, right? If you have a terminal illness, you're coming to the end of your life. This terminal bouton is at the end of our motor neuron. Okay. So we can see here that we have some synaptic vesicles full of neurotransmitter ready to go. So we need a signal telling us to release them. So that signal is our action potential getting down here. This is depolarization. That depolarization, when it gets here, opens a voltage-gated channel, specifically our voltage-gated calcium channel. All right, so this is that same type of channel that we saw when we were looking at the synapse. All right, so it's a voltage-gated calcium channel. So when we have that action potential to get here, we allow calcium to move. Calcium wants to come into cells, so it does so. Okay. So that's our action potential coming down the neuron, releasing calcium into the cell. Okay. And so just like in the synapse, when we get calcium into the cell, we're going to release our neurotransmitter. So we had this neurotransmitter, which we now know is acetylcholine because we're in a specific type of neuron. Right? So we have acetylcholine in these synaptic vesicles. That calcium entry is triggering this release. So we can see this exocytosis here as we have that vesicle dock and then kind of explode outward, releasing that acetylcholine. Okay. So that's all the same as it was in our synapses. Okay? I just got a little bit of specialized terminology here with that terminal bouton. So now we have acetylcholine in this space between our motor neuron and our muscle fiber here. Okay. So now that we have acetylcholine, that acetylcholine needs to go somewhere. So our muscle is going to catch it with a wrist. So here on that muscle side, we can see those nicotinic cholinergic receptors. Right? So we can see the acetylcholine coming in, opening that ion channel. Right? So that ion channel, when it opens, allows ions to move. We know that like elsewhere in the body, sodium really wants to come in. There's a big, strong electrochemical driving force on sodium. So when this ion channel opens, we'll have a little potassium escape, but on the whole, waves of sodium coming in here to our muscle fiber, right? So that's causing depolarization here in the muscle itself, okay? So in this motor end plate, which is what we're calling, right? This part, the motor end plate, is this kind of foldy looking part of the muscle fiber where we have the connection to the nerve, right? We are triggering depolarization. So this depolarization is our end plate potential, right? So end plate potential from motor end plate is always depolarization, always excitatory. So this is like our EPSP in the synapse. That we don't have to decide if there's excitatory or inhibitory potential here, always excitatory. EPSP always is depolarizing our muscle fiber. Okay. So this causes an action potential in the muscle fiber itself. So we have a wave of depolarization crossing the muscle fiber. And as we talk about what's going on in muscle cells, we're going to think about how that actually causes the muscle fiber to contract. So we're not doing that yet. But know that we have this depolarization, we have this action potential, and ultimately it does cause your muscle to contract, meaning your muscle gets shorter. That's really what's happening when you use a muscle, is the muscle gets shorter in length. And in order to relax our muscle, we've got to get rid of that acetylcholine. We've got to get rid of this signal, right? So we have an enzyme, our acetylcholinesterase, that breaks down acetylcholine, right? So now we don't have acetylcholine to bind into the receptors. We have some reuptake molecules on our motor neuron that pull the choline part back in so that we can make more acetylcholine. And this is, in general, our whole kind of process here for triggering contraction in a muscle cell at the neuromuscular junction. I know that's a lot of words, a lot of pictures, and a lot of lists. Questions about this? Yeah. Questions about this tomorrow? What's going on in video form? So you can see this
nerve impulses,
quote unquote 3D
action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal.
I don't want to keep you guys too far over. I'm going to put this on double time so you can see the picture. They're the same steps listed on your slides. And as you can see, they're on YouTube.
Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay. So next time, our review of this will be talking about poisons real quick, and then we'll break into muscles themselves. Have a good rest.